Yes, so my capstone was working with the construction of a probe for the identification of phosphorylation-dependent binding partners of beta-Catenin. So I'll start by just simply talking about what beta-Catenin is. Beta-Catenin is a protein in the Wnt pathway. So the Wnt pathway is a highly regulated pathway that is involved in growth and development. The Wnt protein binds to the frizzled receptor, and that turns the pathway on. When the pathway's on, it inhibits the destruction complex and allows beta-Catenin to accumulate in the cell, and it operates as the transcription factor for a variety of genes involved in growth and development. The destruction complex, when the Wnt pathway is turned off, and the Wnt protein is not bound to the frizzled complex, into the frizzled receptor, the destruction complex, which is made up of APC, axin, and two kinases, GSK3 beta and CK1 alpha, phosphorylate beta-Catenin at 4 different spots on the N-terminal region. These phosphorylations, specifically 2 of them, are what allowed β-TrCP, the substrate recognition protein of the SCF ubiquitin ligase complex, to bind to beta-Catenin and polyubiquitylate it. This is a tag that allows the proteasome to degrade beta-Catenin. When beta-Catenin has mutations in that N-terminal region and prevents those phosphorylations from being phosphorylated, it prevents β-TrCP from recognizing it, prevents ubiquitination. It accumulates in the cell and upregulates genes such as c-Myc, VEGF, and BIRC5, which again are involved in cell proliferation and growth, which often leads to cancers. Beta-Catenin is associated, mutations in beta-Catenin is associated with cancers such as liver cancer, colon cancer, uterine cancer, and skin cancer, as well as multiple others. And mutations in the Wnt pathway, including commonly with APC, are found in about 10% of all cancers. So understanding the mechanisms of the pathway and understanding the function of the phosphorylation sites on beta-Catenin is of great significance. So my primary goal was to design a chemically defined probe of phosphorylated beta-Catenin to investigate its interactome, or the proteins it interacts with, and we wanted to do this in vitro. And then the aim 2 was to identify the proteins that it interacts with or engages with the N-terminally phosphorylated beta-Catenin, and our method was an in vitro biotinylation assay, or BioID. So biotinylation assays, or BioID, are typically done in vivo, and you would transfect mammalian cells using a complex of your protein of interest, which is bound to a biotin ligase, to tag proteins that it interacts with biotin. And then you can analyze those proteins either using Western blot for initial analysis or mass spectrometry for identification. So why did we decide to use in vitro methods instead of using our cell? The primary reason is that when you use in vivo methods, you have a protein and it's phosphorylated, but you don't know exactly where it's phosphorylated. You don't know for sure that it is phosphorylated. And so our in vitro construction allows us to know precisely which residues are phosphorylated and that they are certainly phosphorylated. So how did we construct this probe? Our probe is made of 3 parts, essentially. We have beta-Catenin, which is our protein. We have our biotin ligase, which is a promiscuous biotin ligase. It can biotinylate any protein. And then we have our phosphorylations. We have our modifications. So the way we made this is we expressed beta-Catenin and our biotin ligase as a fusion protein in E. coli. And then we synthesized a phosphorylated peptide that had 2 phosphorylation sites on it, and then chemically ligated that peptide to our fusion protein to produce the phosphorylated beta-Catenin probe. So we can see in this gel that we have beta-Catenin probe. We attached a fluorescence marker to our peptide in order to track and determine that if we had properly ligated it to the peptide. You can see in our fluorescence image of the gel that we did indeed have ligation of our peptide. So we made our probe. We made our probe, and the question is, does it work? And how can we best tell if it works? So the first step we wanted to do was to get as clear and simple data as possible. And we know that from previous research that β-TrCP should interact with the phosphorylations that we have on the N-terminal region. So our first attempt was to express β-TrCP in vitro so we could run a biotinylation assay of the two together. We started by trying to express β-TrCP as in wild type or unmutated form, and it quickly and easily precipitated out of our solution, indicating that it was essentially insoluble. So doing some more looking into the literature, we discovered that people had reported it was easier to express and more soluble if you truncated a large portion of the N-terminus, as well as co-expressing with SCIP1, which is a protein that it binds to and would allow it to be more soluble. So I designed primers. We did a modified version of quick change PCR to mutate the N-terminal region, as well as to delete a couple disordered loops in SCIP1 and to co-express them. And when we co-expressed them, we kind of got unclear results. Our anticipated weight for our protein with β-TrCP with the tag that we had a GST tag on should have been 77 kilodaltons. This is a gel from our size exclusion chromatography fractions, and we do have a band right around 77 kilodaltons. It's lighter than some of the other bands that we have, and so if we have β-TrCP, it's certainly not very pure, and it's at relatively low concentrations. Ultimately, we determined that insect cells would have probably made a better expression host than E. coli, but at this point, it was a little too late to express an insect cell. So in the future, if we want to express, we'll definitely use insect cells. So using what we had, which again, may or may not have been β-TrCP, we did an initial biotinylation assay. So we used two different concentrations of biotin, and we used a phosphorylated version of our β-Catenin and our unphosphorylated version. And we didn't see any band for β-TrCP, which isn't too surprising considering if we had it, it was at a very low concentration, and it's not even clear if we had it. But what we did see is we saw auto-biotinylation, or our biotin ligase was biotinylating β-Catenin or itself, potentially. What was kind of concerning about this data, though, is that we only saw auto-biotinylation for our control, our unphosphorylated version, and we didn't see it for our phosphorylated β-Catenin at any time point or concentration of biotin. Which raises some questions. Is our peptide somehow inhibiting auto-biotinylation? That doesn't seem likely. It's possible maybe that it was actually interacting with β-TrCP and that was reducing the amount of auto-biotinylation, but we didn't detect it. Or maybe somehow our biotin ligase enzyme was inactive. Those are all the thoughts kind of running through my head. We ran a gel of our reaction aliquots, and you can see here that the concentration, the intensity of the band for our phosphorylated version was substantially lower than the concentration for our control, which is a problem because that was just the amount we're adding. And so it should have been equalized. When I reran a concentration gel and quanted it to determine the concentration of our aliquots, I discovered that we had probably about 2.5 times less phosphorylated protein than we had of our control protein. So that was likely the reason we didn't see auto-biotinylation on our Western, is just that we had way too little of it. Unfortunately, we weren't able to update our concentrations in time before we ran our next assay. We ran our next assay using HEC-293 cell lysates. And so this is kind of to look at a full screen of exposing it to all the soluble proteins in a cell to analyze and see if it interacts with any novel proteins and see if we can see any difference between phosphorylated beta-catenin and unphosphorylated beta-catenin. So the left side of the Western blot is our phosphorylated beta-catenin, and the right side is our unphosphorylated beta-catenin. at different time points. And we also still wanted to have a background of being able to see β-TrCP and interaction with that as a positive control. So in half of our HEC293 cells, we had transfected them with β-TrCP, and the other half was our control. And what you will notice is that for β-Catenin, we do see autobiotinylation. And of course, it's higher in the unphosphorylated β-Catenin than in our phosphorylated β-Catenin because we have a higher concentration of that. We also have gap pH in fluorescence screen as a loading control for our cell lysates. We do see a band at the correct molecular weight for β-TrCP, and it's not entirely clear that this is for sure β-TrCP. It seems likely, and there's some reasons for this. The one is that when we transfect with β-TrCP, we have a higher concentration of, or higher intensity of our bands. That's the strongest indication that it is β-TrCP, in addition to being at the correct molecular weight. There's some interesting aspects of the data, though. This is, again, first pass preliminary data, so we haven't run additional experiments yet, so it's very hard to tell with certainty. But you'll notice at time 0, we have what appears to be biotinylation in our β-TrCP. So that raises some questions. If at time 0 we have it, is it an endogenous biotinylated protein? And one thing that I'll note is that if you look at our unphosphorylated β-Catenin, time 0 in the control, you can see it most clearly there. There is some autobiotinylation at time 0, which would indicate that our quenching wasn't necessarily sufficient to properly prevent any biotinylation. So there's likely some level of background biotinylation that we might be detecting as well. We don't see a time-dependent increase in β-TrCP, which is interesting. Our time points of 0 hours, 2 hours, and 4 hours are pretty long for β-TrCP at the concentrations that we have it at. So maybe having shorter time periods would show a time-dependent increase, but it's also entirely possible that even just the background levels are essentially saturating it. And so we don't see an increase beyond time 0. One of the benefits of using biotin as a marker is that there's only 4 proteins in mammalian cells that are biotinylated naturally. One of the downsides is that 2 of them are very close to the molecular weight of β-TrCP. So β-TrCP here should be about 69 kilodaltons, and 2 of our other proteins are at 72 and 75 kilodaltons. So it's really hard to say at this point what we're looking at here. But, so what's the takeaway from all of this data? And I think the takeaway from this data is that we have created a chemically defined probe, and it is enzymatically active, right? So my worries about the biotin ligase somehow not being active is clearly shown to be false here. We have autobiotinylation in the phosphorylated β-Catenin, which clearly indicates, even at relatively low concentrations, there is detectable activity. So what are my conclusions and our next steps? We have produced an enzymatically active probe for identifying the interactome of N-terminally phosphorylated β-Catenin, and it's chemically defined. We know precisely which residues are phosphorylated, and given proper reaction conditions, this method should be effective for detecting novel binding partners of β-Catenin when it's phosphorylated. So our next steps are to run more biotinylation assays. That's our very first next steps. The very first thing we need to do absolutely is equalize our concentrations of our control and our phosphorylated protein. Other things that we could probably do is to lower our concentration of biotin. We are operating substantially over the km of biotin for our biotin ligase because we wanted to make sure we saw some results. But reducing that would lower the likelihood of seeing saturation, and it would probably help increase the difference between phosphorylated and unphosphorylated. We should make sure that we have proper quenching controls. We didn't use some controls in order to preserve our aliquots of cell lysates, but then it made it a little harder to determine what we were actually looking at. And if we can possibly decrease autobiotinylation, that would also help in terms of being able to see other proteins that are present at generally lower concentrations than beta-catenin and detecting biotinylation there. So acknowledgements, Hannah Crayley was the master's student I worked with most closely on this project. And then the other members of my lab, Nader is a collaborator who provided the cell lysates, and Beth Bennett is my advisor, and then Daniel Dempsey is my PI. So I will open it for questions.